Well, like chilling right now. History, my BP was history lecture. It was in 2015 and 2015 to the week and both that.
Yes.
I just to remember something and like inspiration and
Oh, it's like,
and I'm like, shoot.
it's just,
And it's been really cool. Such a bit in your walk since being up there garden.
yeah,
Have I the flown? What can you do about yours?
just,
Sleep.
on to whatever, Mr. Activate.
I think we felt like, that's back.
Correct. So maybe
So,
this, I want a couple more pride.
I'm not.
And horror. Next.
Now, maybe because often
I could, it's Good Friday. Well, it's, celebrate. Yeah, watching. So, I'm assuming that's your word sign.
so 100%
Records, yeah, he's making sure. I'm going to go for like a safe. It's others fucking work stuff. When were the years come teenager, my parents left. So they went to visit them sister, but I had the soon spell on the irrespective. So he left me at home to we went to listen to some bread nothing. So on Friday I was like, okay, great. I was like direct to McDonald's and grabbing the business. And as soon as I got there, I realized the more cost of trying and I need to say so I happened to like I had like one pulse walk and so I kept like sale McDonald's fries that put away. I said that
I think fine staying like that all the time.
no, I think because everyone was out in town, this was a show. So not because the ball no one's here after the pieces.
And it would
Don't directly.
did you like
Listen.
and I was like I went up I was trying to cigarette and I was like for magic
I'm stay right now. Oh, my gosh.
that'll be on first one.
It was.
I thought so and that's a good unplugged.
You're starting to think that. Yeah, it's just my dad was so.
That's a never there.
So that was all
Sure else
and maybe like kiss God and it about this precious stone to the sunshine. Please, no.
how is that's a fan thing Andrew.
Okay, but don't say actually. It's like it makes it seem like everything everything.
So it's not the parent that was done. It's just like I always think about this thing.
Yeah.
I was like something else like it was like
They kind of stay in college.
and that they call it we did on you're catching the stokie.
But they're still plus the race.
When man out of the things.
If we have a side next,
Now look man. Nothing
I mean, to be fair, like, then I'm thinking, like, maybe there's an usher would have parts like being able to product, Chad. How I got to the project, tried down this, I couldn't, it's good, that case fishing. I doubt this is not project, and the project's hard, rather soon. Man,
Welcome everyone. It's my pleasure to introduce Sriram Sankararaman.
yeah.
But at the end, sorry. I met Sriram when I was on Swapska, and he was going PhD students. All the same 98, but it, years of PhD skill. Yeah, he's now a professor of computer science, human genetics, and computational medicine. He has done a lot of work that spans both human genetics, CS, and population genetics. So he was interested in lots of things, which I think it's this combination. And this work is really interdisciplinary, which I think that is pretty nice. And within the field of computational biology, it is really interdisciplinary. And so that I'm gonna let him tell us about his latest exciting research. That's our thank you for doing.
Okay, thanks for that the invitation. It's comedian. It's been great meeting everyone here. And so today I'll tell you a few different lines of work that we've been pursuing in my lab over the past few years. So when I asked him, what should I talk about? And I gave her two possible topics and I will potentially talk about. One is focused on archaic ancestry, so that's for pop gen.
and others focused on complex traits. And we said that we people interested in both, so I should try to fit both in. So that's what I tried. So it's going to be an admixture of these two. I'm tracking this. And feel free to interrupt me at any point because some of this might be fairly hyper. So happy to take questions. Okay, so broadly, I'm interested in genomic data and trying to understand genomic data using tools from statistics and machine learning. And there are a few different themes, as I said, that I'm interested in pursuing. So one set of topics inspired by the fact that UCLA has access to a great hospital system, which is recruiting patients and trying to improve clinical decisions, is how do we use genomics, machine learning, statistics to improve treatment, improve clinical decision. I won't say too much about this line of work, but in today's talk, I'm going to focus more on these other two lines of work. So one is on understanding evolutionary processes from genomic data, and the second one is in understanding the relationship between genomics and what we call complex traits. So in the first part of my talk, I'll focus on the link between genetics and evolution. And a lot of this is really focused on understanding the relationship between archaic and modern humans. So essentially, these are populations that have been inhabiting the Earth over the last hundreds of thousands of years. And based on genomic analyses of both present-day and ancient populations, a lot of details of the interactions between these populations are now being made. For example, based on modern human genomes alone, we know that there was modern humans evolved in Africa around 300,000 years ago. And about 100,000 years ago, there was this migration of Africa, and these individuals were the ancestors of present-day non-African populations. On the other hand, based on the fossil record, we know about other archaic hominins. The most prominent amongst them are the Neanderthals. And we know that in the fossil record, Neanderthals appear again about 300,000 years ago. And they disappear from the fossil record about 30,000 years ago. And in terms of the geographic span, these fossils are found across Europe and Central Asia. And so modern humans and Neanderthals overlapped both space and time. And so this kind of picture essentially leads to questions about what was the interaction between these populations and what were the consequences of these interactions. And by and large, these were questions in anthropology, in archaeology, but all of this changed around 2010 when we really got access to the first genome sequence from a Neanderthal. And that was, in some sense, the start of the field of ancient DNA. And since then, there have been many such ancient genomes that have been sequenced, and we're really comparing genome sequences from present day and these archaic populations. We're able to tease apart the relationships of these two genomes. So, for example, this is a picture that from a paper almost 10 years ago, which looked at what we call admixture events, interbreeding and intercrossing events between different modern human populations and Neanderthals, as well as Denisovans, which are another archaic group that is closely related to Neanderthals. The Denisovans are particularly interesting because these were a group that were identified primarily based on genetic data. We didn't have much based on fossil evidence to initially identify and characterize these groups. But the key takeaway is these arrows in this kind of evolutionary tree, which point to these admixture of intercrossing events between archaic and modern population. Beyond these kind of arrows which hint at global events, we are now also able to identify regions of the genome of present-day humans that trace their ancestry to these archaic populations. So we are able to build these maps of Neanderthal, Denisovan ancestry, and essentially get pictures that look like this, where you can quantify what fraction of the ancestries of different populations trace to these archaic events. So that's about the demographic processes, but we can also now start studying what was the biological consequences of these archaic admixture events. And a lot of this has to do with using these maps as a starting point. So the maps essentially tell us in a particular individual's genome which regions trace their ancestry to Neanderthal routines. And then you can use these as essentially genetic variants to then correlate them with different aspects of fitness or phenotypes. So, for example, some pictures that emerge are there are some regions of the genome where there is much more archaic ancestry than you would expect if these regions were mutually evolving. So, in the context of Neanderthal retrogression in the ancestors of non-Africans, there are regions like that. So, that overlaps a gene called BMC2, which are at a frequency of close to 50% today in present-day Europeans, compared to about 2%, which was the frequency of Neanderthal ancestry about 50,000 years. And additional modeling has shown that these types of regions likely couldn't have arisen to these frequencies by chance. So likely there was an adaptive reason why they had the frequencies that we see them. Beyond this, if you look more globally, the picture is that there seems to be some deleterious consequences to the archaic regions. For example, if you look at regions of the genome which are known to be constrained by selection, functionally important regions, in general, Neanderthal, as well as Denisovan ancestry across different populations, tends to be lower in the selectively constrained regions rather than in neutral regions. So this suggests that there is a kind of a cost, a deleterious cost associated with archaic alleles on average. And beyond just looking at fitness, you can also interrogate specific complex traits. And so this is how I got interested in the relationship between introgression and complex traits, where we can ask what is the impact globally of these introgressed, say, Neanderthal alleles on different types of complex traits, and how it compared to a non-introgressed allele. Isn't the case that on average, these introgressed alleles contribute more than you would expect, or is it less than you would expect? And in this analysis from a paper a few years ago, what we find that is systematically across many different traits that we looked at, introgressed Neanderthal alleles tend to contribute less than you would expect. So we call this how much heritability comes from these introgressed alleles. And in general, there tends to be a depletion of heritability. And that is consistent with the fact that these alleles tend to be deleterious on average. So this is all building a picture of what we think is the consequences of these archaic introgression events. But everything I've talked about today focuses primarily on Neanderthals and Denisovans, and that is a consequence of the fact that these are the archaic groups from which we have high quality reference genome. So the question that led us to start thinking about the study that I'll tell you about is what do we know about admixture in populations in Africa, where most of these prior work, going back to work by Ward and colleagues, suggests that there exists these deep lineages. However, we don't have archaic genomes to be able to pull out and study in detail these types. And so the question is, what can we learn about these kinds of events in the absence of high-quality ancient genome? So to study this, we have to develop different kinds of statistical models. And I'll tell you about one line of evidence. There are additional lines of evidence that we've been working on and we've published, which try to help us figure out what might be the role of admixture in the ancestry of African population. So the key line of evidence is using this very powerful tool to study the history of populations. And this is looking at genetic variation and what is the frequency of different genetic variants. So this is a tool called the site frequency spectrum. Just want to make sure I'm going into have detail. How many people know what a site frequency spectrum is? Okay, a lot of you, but so let me explain what a site frequency spectrum is. So you have a collection of genomes, so each individual is among the rows here, and you're looking at positions where there is variation across individuals. So each column here is a variant. And what a site frequency spectrum does is you go to the first row and you ask what is the frequency of the variant, either the derived allele or the minor allele, depending on which version you're looking at. So in this case, the derived allele will be the A allele. So it's derived relative to, in this case, the chin. And so it is at a frequency or a count of 2. So now you go to the next position. And here again, the derived allele is T allele. So here again, you have another site where you have a derived allele of at count. So you make a histogram of all of these different sites. So the histogram will be what are the frequencies of the counts on the x-axis, and essentially it's a histogram of their frequency. And what is interesting about the site frequency spectrum is it can be an incredibly powerful tool to understand the demographic history of this population. So there is population genetic theory that says under certain conditions, essentially this is a sufficient statistic. All the information about the demographic history can be derived from this statistic. Now, what we are interested in is in using the site frequency spectrum to learn about archaic information. So the problem is the site frequency spectrum by itself, because it is influenced by all aspects of demography, has too much information. So we'd have to model every kind of detail of African demography, including potential admixture events, to really be able to fit the classical site frequency spectrum. To avoid having to do this, we use a modified version of this, which we call the conditional site frequency spectrum that was introduced in this paper from Monty Slatkin's group. And what we do in the CSFS is the following. We have some population that we care about analyzing, and we have an outgroup. So an outgroup could be, say, Neanderthals. And we're only going to compute the site frequency spectrum at those sites where Neanderthals, the output, carry the derived allele with respect to, say, the ancestor, in this case, Chad. So what we do is we eliminate all of these other sites where Neanderthals carry the ancestral allele. We only focus on those sites where they carry the derived. And now we build the site frequency spectrum as before. So this is called conditional site frequency. Now, why is this a useful thing to be looking at? So there is an interesting feature of this conditional site frequency spectrum that was shown in the paper from 2007, which is the following. So let's say you have a population, an ancestral population, that splits into two. So the population on the left is the population you want to study, and the population on the right is the outgroup population. And if you now ask, what does the conditional site frequency spectrum look like in the African population conditioned on Neanderthals in this model of demographic history, the answer turns out to be that you get a uniform distribution. So this is what the CSFS will look like under this demographic history. And importantly, this result holds independent of the history of population sizes in the African population. So no matter whether there were population expansions, contractions, and so on, the conditional site frequency spectrum has this shape, which is uniform. So that's a nice feature to have, because essentially it tells us we can ignore some of the complexities that are happening within this African group. And now we can use this to study what the CSFS looks like in real life. So this is the idealized setting. Now the question is, what does the CSFS look like when we look at an African population, say the Yoruba population from 1,000 genomes conditioned on, say, Neanderthals or Denisovan Spreader. And when we do this, we find something quite different from the film. You find this shape, essentially this kind of U-shaped conditional site frequency spectrum. So this is in Yoruba, where they've conditioned on either Neanderthals or Denisovans. That's the one. And now we looked at other populations. In this study, we mainly looked at West African populations, and all of these populations have this basic feature of the CSFS. So what this is telling us is clearly this model, where there was a clean split between African populations and Neanderthal populations, is not something that fit with the data. So what might explain this? Of course, there can be technical artifacts. There could be errors in our genotypes, errors in the archaic genomes. We actually did some calculations in this study. We found that error rates would have to be really quite high to be able to explain this kind of a feature. So this cannot just be explained by errors in the data. There are other processes. For example, all of this result assumes alleles are evolving neutrally. So what happens if that's not the case? So we looked at examples where the population, most of the alleles are evolving according to monotonic selection of different kinds. And again, we found that that could not. So this has to be something which is a demographic process that it's great. So what might explain it? We looked at a few alternatives. So the first one is perhaps there was some kind of introgression event from the ancestors of Neanderthals into the ancestors of African population. So there's not a clean split. There was some introgression following this initial split. And so when you model this, so this is a model which has some additional introgression events, but you have this introgression event related to the Neanderthals. You find that, yes, it deviates from uniformity, but it doesn't completely reproduce what we see with the seed. And primarily what we see is you only get an increase of alleles which are low frequency derived. You don't see on the opposite end, the high frequency derived genes also depends. So Neanderthal-related introgression doesn't offer a full explanation of this ancient of the data. Another possibility is, again, instead of there being a clean split between African population and Neanderthal populations, maybe there was a split and there was some kind of structure within Africa. So within Africa, populations split, come back together, and then they mix, and this happens over a period of time. So we can ask what happened under such a model, or what we call it deep structure within Africa. So in this example, you have a split between the population leading to Africa and the Neanderthals, and then you have this deep structure. You have some splits followed by some migration. Turns out now you can see something that looks like a CSFS. So you do see it happening on both ends, but it doesn't quite fully explain the CSFS. And the reason for this is in the actual data, these types of models can only produce something which is completely symmetric. So the CSFS is always going to have this symmetric shape. What we see in the data is actually not symmetric. It may not be obvious, but it turns out that the high frequency end is always higher than the low frequency end. So it doesn't give us a good enough fit to the data. So what else might cause an explanation that looks like this? And it turns out one model that does explain it is the following. So now you have introgression into the ancestors of different populations from a lineage which split off prior to the split between all-influence and Neanderthals. So this is a qualitatively different model than everything else we have talked about. So the first model was it must be from something that was related to Neanderthals. The second one was it was two populations closely related to each other, mixing. And now you have a population which was even further back in time. And now you can, with this type of a model, so we call this a model of archaic introgression from an unknown lineage, you can actually begin to explain the scenes there.
Sorry, Brady, are you just only talking about the Neanderthal model, but also testing the Eastern model? Because, yeah, I guess the CFS at the conditional psychiatry spectrum also looks very skewed when you consider that it's asymmetric. So I was just wondering if it's actually incorporating your model, but it's not talking about it right now, or...
Yeah, so there are two aspects. So yes, so in our model here, Neanderthals and Denisovans are essentially sister groups. So this kind of an assumption that we're making that there was a group that split prior to the modern human Neanderthal split, essentially would mean that it's also applicable to the Denisovans. There is some complication which is that was called the super archaic contribution to the Denisovans, which you might be thinking about. Yes, so that what it does is it causes maybe a slight difference between Neanderthal and Denisovans, because super archaic contribution is a fairly small contribution to the Denisovan genome. So it can change things a little bit, but beyond that, they are essentially identical as far as this model is concerned.
Would you mind going back to the setup for the conditional site frequency spectrum? Yes. So you restrict yourself to sites in which the Neanderthal differs from the chin.
Differs from the chin, yes. More generally, the Neanderthal is derived relative to what you would call an ancestry. Yeah, and it's interesting. I didn't talk about why this is the case, but for those of you who like to think about site frequency spectra and theory, at least I can sketch out the proof, which is actually quite elegant. The idea is the following. So imagine you have a population at the root, and that's kind of under mutation drift equilibrium. So typically, that's what the site frequency spectrum looks like, 1 over x. right? And now you're ascertaining, so you're essentially picking out those sites where Neanderthal is derived. So if things are evolving neutrally, the allele frequency of a site which is at X at the root is going to be on average X in the Neanderthals and X in the Africans. So the probability of picking a derived allele is simply the allele frequency. So the probability of ascertaining it is given by X, which is given. So now the conditional site frequency spectrum is the probability of this multiplied by its ascertainment, and that leads to this unified. So that's the general idea. Of course, this actually holds quite generally, even under deviations from all of this uniformity assumption bonus. And it's almost like a quantitative test, where under this model or kind of deviations from this model, it's uniform, and we're looking for a deviations from uniformity, which tell us A, that this model is also right, but also tell us which of the alternatives might.
Right.
Yeah.
Yeah. And you figure that initialize best along the two mimics target. Right. Good. So here you would feel that conditionally classified as Neanderthal, and then we submit and system.
Yeah. So the two, so the blue and if you can see the red, the orange. The two blues are empirical CSFSs, where on one hand you're conditioning on the Neanderthal, and on the other hand you're conditioning on the Denisovans. Right? So you can see to a large extent they overlap. We have some hypotheses of why there might be some deviations, but yes, the first order thing, yeah, basic. Yeah.
My last question is for like the Denisovans, since we only have one, we have to, I guess, assume that this is representative to some degree in practice. But for the Neanderthals, I guess in 2028, there was at least two, I think all the charts star was published by them. Did you use both or like in conjunction and then essentially conditioned on now, like that the drug will how to be fixed across 4 instead of 2 chromosomes?
Yeah. So if I recall correctly, what we did is, so we have the two Altai and the Median Neanderthal, we just conditioned on one at a time. So we had in the paper, so we might have actually looked into different Neanderthals activity spectra. We could do additional things with additional Neanderthal genomes. It could actually improve the precision with some of the modeling. It won't change the qualitative results, but it might actually change how precise some of these results are.
Yes.
Okay, so now you can essentially use this as a statistic, right? And you can, so essentially we're capturing the gross features of the statistic with a model like this. Now you can actually parameterize this model with a few different parameters and ask what set of parameters would explain statistics. So it's an approximate Bayesian computation approach that we use, and you can come up with some numbers here. I will say at this stage, these numbers have wide confidence intervals, so I wouldn't interpret them too much. But the main kind of feature here is this lineage that split off prior to the human Neanderthal split and contributed genetic variation in specific pathogens.
NPS and 43K is the mixture.
43K is the mixture time, and this unpublished. So here again, there are wide confidence intervals here. More recent work, what we have done, and I don't have the slide here, is we try to basically get at that by jointly analyzing African and non-African population to at least allow us to figure out could this have predated or post-dated the out of Africa event. And what is interesting is if you look at the non-African population, the CSFS also looks like this, with an additional contribution now coming from the Neanderthal. So that's the difference in the CSFS in non-African populations. And so what that is telling us qualitatively is at least some part of this integration happened prior to the out of Africa event and was shared by essentially all modern human populations. Yeah. Okay. So this is one line of work to try to understand ghost population. In this paper that we wrote a few years back, we looked at another line of evidence, which is looking at trying to pull out segments of the genomes of, in this case, West African populations that trace their ancestry to this archaic intropatient event. Key idea behind that, I would say, is essentially looking for regions of this genome where you have extremely deep coalescence lines that would be suggestive of this archaic intervention event. It turns out we can put out these segments and perform analyses which are similar to this and trying to figure out how much of the region comes from this ancestry, and we get qualitative survey. I won't say too much about that. I will say there's been more recent work which has looked at this question in a lot more detail in terms of modeling. So this is work from Brenna and Simone Carrero's lab. And essentially, they have gone into a lot of additional statistics, looking at many more genomes within Africa, and trying to understand what does the story look like. And as you can see, the picture is extremely complex. So you have all these splits, you have these events. But I just want to draw attention to one aspect of this. So we have two models which are appear to be equally good fits to the data. And both of these models have what is called the STEM population. And the STEM population, if you look at it closely, is essentially this archaic population. So it splits off prior to the Neanderthal split. And so clearly the picture is more complex, but the key kind of takeaway is there are these old population, archaic populations that are contributing to genetic variation. So where does this leave us? So my current work now is really trying to understand how do we make sense of these increasingly complicated pictures that we are drawing. How can we infer them from data? How can we infer them in robust ways? So essentially, we're going from a model of evolution, which is tree-like, to this model where there are these graphs that better describe human history. And so how do we go about inferring these graphs from data? And what is lacking in my mind is phylogenetics, inferring trees from data. There's a lot of theory out there in methodology, which gives us good guidance of what kind of trees we can estimate, what are procedures, how consistent they are, and so on. A lot of it is missing with our ability to infer these classical models of network models. And I'll just say this one anecdote from a paper a few years back. So there are some methods that have been developed to infer these kinds of graphs. Typically what they do is they'll start with a tree, so they'll estimate an initial tree, and then they'll start adding some edges to these trees to make them fit the data better. So add trees that give you better fit, say, a likelihood data. And what we find is essentially those kinds of methods, for kind of technical reasons, they get stuck in these local optima. And that's because the space of graphs is way more complex. You can't use the traditional moves to explore tree space in the space of graphs. And so we can prove that some of these methods are actually probably inconsistent in estimating these graphs. And of course, how do we fix this? We have some proposals of moves that explore graph space better that we talked about. But again, that's just an initial attempt. I think there needs to be a lot more done into studying this problem and coming up with good theory and output. Okay, so that was the part which was on archaic architecture. So now I'm going to switch gears and I'll talk about complex strings. Okay, so a lot of this, the way I got interested in this is actually two different reasons. And the first reason was, at some point, I wanted to figure out what is the relationship between these interconnected alleles and complex strings. Do they have an impact on strings? And these interconnected alleles, typically from Neanderthals and Denisovans, they're of the order of a couple of %. So which means on average, their frequencies are not going to be too high in most human populations. And so to be able to have any power to study the impact, you need large sample sizes. And it was at that point, this was in 2016, that we learned about these biobanks. So I learned about the UK biobank, which was essentially collecting data sets at a scale which would make these kinds of analyses feasible. And as we got interested in answering this question, I realized that there were some more kind of fundamental questions about complex trees that we had to answer before we started looking at. So that was my kind of segue into this topic. And really, I've gotten more and more interested because of the kind of data that's being collected. So we have these biobanks. I have one at my institution at UCLA. There are several such biobanks across the US, Europe, and Asia. And what's happened over the last 10 years or so is there's really been a big change in terms of the diversity of phenotypes that are being collected. It's not just one disease that we're interested in. They're collecting everything that could potentially be measured in a patient or a health system. The other big aspect is the scale at which these data sets are growing. And finally, diversity in terms of the populations that are being analyzed. So you now have biobanks which are trying to have a much more diverse collection of individuals in terms of their ancestries. So all of this is exciting from a data point of view, but that also brings with us lots of challenges. And that's been something that has kept me motivated over the last several years, which is you're going to have these data sets with hundreds of thousands of genomes, now approaching millions. What are the insights we can get from these data sets, put it into genetic basis of complex tree? So that's a very broad question, and there are many different angles to this. One angle would be really about statistical modeling. So how do we actually build models which capture the biological processes we care about that are robust to all the artifacts and biases in the data set? So coming up with more expressive or realistic statistical models. And then you have the other side of this picture, which is once you have your model, how do you actually get this model to compute things on these very large scales? That's an algorithmic question. How do you perform inference estimation in this model at the scale of these very large scales? And oftentimes there's a trade-off. These two are in tension with each other. The more interesting, the more complicated you make a model, you'll have to somehow kind of figure out the computational issues. And finally, there is another aspect which is also really interesting, which has to do with data access and privacy. So how do you actually perform some of these analyses with not that much data being released or across different data sites that don't want to release all of that data? And that's an extremely important question, but I won't say too much about it. So today's talk will primarily focus on these two. So kind of the statistical and computational trade-offs when dealing with some of these data. But before talking about complex traits, there are other kind of challenges where you deal with biobank data. So I just said a couple of slides back, sample sizes are great, but it can be misleading. So it's like an anecdote. So we have a collaborator who was interested in studying some particular phenotype in the biobank. You're excited about the numbers. You have 100,000, but then you look at the phenotype you care about, and quickly the numbers don't, right? And this is, of course, a pervasive problem. Most phenotypes are finally missing. So this is what it looks like in the UK Biobank. And what's problematic is many of these are often most clinically relevant or clinically or biomedically interesting phenotypes. And the reason for this is there are issues of cost and logistics at making kind of gold standard measurements of these phenotypes that you can. So there is, again, a trade-off, kind of a tension here between measuring phenotypes at scale but sacrificing on kind of highly accurate measurements. So the problem is you have a lot of missing data. And so how do we deal with this is something we and others have been grappling. So one issue is you throw out every data point that is not missing. That, of course, reduces your sample size. The alternative is to try to statistically fill in missing data. So this leads to what we call equidation. And the key kind of idea that allows this to be enabled is the fact that in these biobanks, you don't just pick one or you don't just measure one phenotype, you measure many phenotypes. So even though maybe the phenotype you care about is missing, there might be other phenotypes that are related to the phenotype you care about that have been measured. So hopefully we can use that data to impute our missing. So this is the classical example. So phenotype 2 might be missing in this individual, but let's use the fact that 1, 3, and 4 might be predictive of speed and intent. Now, what makes this problem different from standard supervised learning is the fact that what you use to impute those other phenotypes themselves might be missing, right? So what you're really trying to do is fill in all of the missing data together. So this matrix you're trying to fill in all of these different missing entries, and that's the phenotype part. And this is a very well studied, mature problem going back to decades of work in statistics and machine learning. But as we started digging deeper, we found that there are some challenges in getting these approaches to work on the kind of data that we are doing. And so really, the desiderata for us in terms of what we'd like are this kind of an imputation approach. We want approaches that are expressive, so we capture not just linear relationships, but potentially non-linear relationships amongst the different phenotypes that scales to these biobank data sets. And the final point, which is an underappreciated point, is that these methods should leverage the fact that the structure is, even the missingness is not randomly inspired. There's actually some underlying structure that leads to this. And just to give you an example of what that might look like, here is one example of structured missingness. So this is from the UK Biobank. So this is a mental health questionnaire that they administer to the participants. And here you have some question that somebody answers. And based on the answer to that question, you have follow-up questions that have been asked. So if a person answers either no or does not answer the first question, so they're missing for the first question, then the follow-up question that will be this. So missing is here has structure that we have to actually pay attention to if you want to know effective imputation. So all of this led us to think of a method for actually doing imputation in these biobank data sets. And the basic idea is quite simple. So we have a deep learning model. So this is a model that's called an autoencoder. It takes your phenotype vector for one individual. Some of those entries are going to be the same. The encoder maps it to some hidden layer. Usually it's a lower dimensional vector. And then the hidden layer is mapped by the decoder back to the original observation panel. And when this mapping is done back into the original observation space, you also end up imputing or filling in the missing entries. So this is a very standard approach, worked well in lots of settings. But when we are actually trying to learn this model from data, we wanted to pay attention. We wanted to actually care about entries of missingness that are realistic. So these are not random missing entries. So going back to an example like this, so here we have phenotypes 2 and 4 that are missing. So we basically want the model to observe that phenotypes 2 and 4 are going to be likely to be missing. So it should be able to predict them jointly from the non-missing entries. So to do this, we use this approach, which we call copy masking. So essentially, we simulate missingness and get the model to reconstruct these simulated missingness. But when we simulate missingness, we actually use the empirical patterns of missingness that we see in the data. So in this example, the fact that 2 and 4 are missing would mean we would preferentially mask 2 and 4 and ask the model to deconstruct these entries from the remaining entries. So as a result, you're actually looking at patterns that are likely to occur in this. And now all of this goes into a loss function. So the model is now trying to reconstruct these phenotypes and compare it to the originally observed phenotypes. And now we got this model from the reconstructs. So now we applied this model in simulation. So here were 2 simulations that we did in the UK Biobank. So these are different types of collections of phenotypes that were curated by collaborators. One is focused primarily on cardiometabolic traits, and others on psychiatric disorders. And what we are doing here is we are varying the amount of missingness in the data, and we are measuring the accuracy of imputation. So higher here is good. Of course, as the amount of missingness varies, all methods, their accuracy decreases. But what we find is overall, autocomplete is more accurate than other approaches. So these other approaches, actually the one that does really, really well is a linear low-rank approximation approach called soft impute. And so we think the gain is primarily because of both the non-linearities and the copy masking approach. We also looked at some deep learning approaches that don't do as well. Again, we think partially because of the fact that they're not trained on structured missingness. And we also looked at some more classical approaches like nearest neighbors and mice and sparks. So here is kind of a head-to-head of what imputation accuracy looks like across the different phenotypes. when we look at our method compared to soft encrypt. In general, there is an increase in accuracy across most phenotypes. Of course, that's not universally true. There are some phenotypes where accuracy decreases.
Can I ask about had the masking techniques that you did and how that might I guess how that plays with balance issues in your data? Like how do you kind of combat that? There were like massive overrepresentations of some like factors of your data, but then your model referred and not but like if you counted for what you know.
Yeah, so I think what you're referring to maybe is the fact that if you have a phenotype that is highly missing already, and additional masking would essentially kind of remove all information about that phenotype, and that could kind of change the distribution. Yeah, so the method, I think all methods, they do need some minimum amount of all observations to be successful. So when we actually look at the imputations empirically, we have standard expectations. You can't go below a certain missingness percentage. That's going to be very data set dependent. In this data set, we can go down to maybe about a couple of percent and it still works because it's still a large sample size. It's 300,000 individuals. But if you were to work with a smaller data set, those kinds of thresholds might be. The other aspect of this is, in some ways, the problem you're trying to solve is actually a very difficult problem even evaluating missingness. And the reason is the following. When you start doing copy masking, you've essentially perturbed your data. Right? The data you're looking at is no longer the original data set. Right? So if you really want to measure how accurate a method is, we have to look at accuracy as kind of missingness approaches in its limit of 0%. That's really the point that you care about. It's hard to do because you have very wide standard errors, but at least these low percentages of missingness kind of tell us approximately what we should expect at behavior of each methods. Okay, so we have these imputed phenotypes, and now this is where the fact that this is a biobank design where the phenotypes are paired with genetics turns out to be extremely harmful. So now what do we do now when we have these imputed phenotypes? One question we can ask is, can we use this to improve our power to make genetic discoveries? So for example, power from a GWAS. So in this particular example, we had collaborators who were very interested in this phenotype, lifetime MDD, major depressive disorder. It turns out it is originally observed in only about 20% of the UK biobank, missing in 80%. So if you were to run a GWAS in the original lifetime MDD, to get a GWAS plot that looks like this, so every dot is a slit, things that are high up here are significant associations. Turns out there is one genome-wide significant association that you discover. Now you do imputation and you get a plot based on GWAS of the imputed phenotypes. And so the number of observed associations, significant associations, goes up from, in this case, 1 to 20. So in this case, it leads to a substantial decrease in the number of genetic factors associated with that content. I will caution here that this by itself doesn't mean that the imputation is working correctly. It's entirely possible that a lot of these that we find are biased, that this could be actually noisy versions of the phenotype that we're putting out. So in this work, we actually took these hits and tried to see if they replicate in external data sets, which were not used here. And we find that many of these new discoveries do replicate. So that's giving us kind of post-hoc confirmation that the imputation is working as intended. But that is not a given in any particular study or application of it. The other thing I want to say here is this approach is very gentle. So the autoencoder was one specific choice of a deep learning model. We could use any other model that we care about. The copy masking we do is one particular choice of structural missingness. We did it because we don't know too much about how missingness came about. There are instances like survey data, questionnaire, where we actually know a lot. And those could lead to better ways of designing these copy maps. So I think there's lots of interesting things to be done here in terms of thinking about the imputation model and the model of missingness and how that can be used for better missing data application.
A couple of questions. So you mentioned that you did some validation on the data sets. Would that also with include phenotype data or with the complete report that
Yeah, so those were phenotypes. Those were studies that were done prior using regular measures of, in this case, MBT. So what we did is we would take these hits that we computed and ask what is their effect size, what's the p-value in those other data sets. So that's one type of analysis. We can also go beyond just looking at the top hits, and you can ask across the genome, what does it look like? Is the imputed phenotype genetically similar to another phenotype? So the other phenotype comes from, say, the PTC consortium's depression summary statistics. So you can compute measures like genetic correlations and ask whether the genetic correlations are close to one, suggesting these are similar phenotypes.
Right. Now, one other, sorry, I have a question about the data. Do you need it to be the case that individuals who have missing data that the most ever interested in, their phenotype values are related to the fact that it is thin? So, just for example, I imagine for a lot of social variables, people choose not the yes or this form, they might be coming from a different distribution of the state than think of it as to change the answer. Is that an issue thing?
Yeah, so this is like a missingness mechanism question, right? So is it in kind of this terminology, missingness at random, missingness not at random, MNOT, which is getting at that, right? Yeah, so we don't have any theory to show that this works. However, empirically, this seems to work better than prior approaches. And I'll give you like an empirical anecdote. Again, there's no theory to suggest that it should work. So one of the phenotypes is something like this one, bilirubin, which is a blood lab that gets measured. And it turns out the way this lab measurement works is you have some device, and if the measurement in the device is below some threshold, it gets marked as missed. It's a classic abduct, right? You can impute those, and when you actually impute those, you find that the imputed phenotypes have values which are consistent with low measurements of this enzyme. Right. So again, that doesn't mean it works in every case, but it does seem to be picking up things which are suggestive of missingness not at random. And again, this goes back to you could also decide or copy masks to actually try to account for that. So you could actually get your model to be more sensitive or to pay more attention to those types of missingness.
And
So just to come up, so are being checked, maybe if there are some traits there, and they done and is it like type of trait?
Yeah, so if you look at this, like cardiometabolic versus psychiatric, in general, psychiatric disorders seem to be harder to impute compared to the cardiometabolic tools. There's some confounding here because in general, there is more missingness for the psychiatric traits than the cardiometabolic traits. So clearly more missingness will make things harder. Beyond that, whether there is something inherent about the correlations amongst the traits, that's not something that, they would think. And there's another interesting question here, which is, this is all assuming you have some expert curating a set of traits and they're running imputation. There's a question, what happens if you want to just run imputation on all traits of that? Okay, you can definitely try that. What's interesting we find is doing that doesn't hurt the accuracy. So we have done some experiments where we've gone beyond this and added additional traits. It does appear the accuracy seems to plateau and not increase, but it doesn't seem to hurt it. Again, that's just empirical. Oh, and I can just say this is ongoing work where, as I said, you can throw in different types of models. So one type of model that many of you have encountered in different applications are transformer-based models, which have a better representation capacity compared to autoencoder-based models. And you can also do additional things with these. For example, you can look at things like the column names might tell you something about which set of features are related to each other. So you could try to leverage that additional piece of information to do better imputation. So we have been trying this, and this method seems to be doing quite a bit better than our and other approaches. So it looks like, again, as you can see, on an absolute scale, imputation accuracy on the whole is still quite low. So there is a long way to go to get accurate imputation on these large data. Okay. How am I doing at the time? Proceed.
The 5 more minutes.
Okay. To get to the part at the top, which I want to focus on. So, okay. So let me just talk a little bit about the thing that I am most excited about, which is understanding. So let's say we have traits, we have genotypes, and we like to figure out this relationship between traits and genetics, which we call genetic architecture. So it's really all about figuring out this mapping from genetics, environment, and trait. And really what we mean by this is we write down some statistical model. The model has parameters, and those parameters have biological interpretation. So we can ask how much of phenotypic variation can be explained by genetics. So we call it broadly heritability. How are genetic effects modulated by context? Depending on context, that leads to quantities like dominance, gene-gene interaction, gene-environment interactions, or how genetic effects are shared across phenotypes. We call that genetic combination. So to make this completely clear, let's look at the simplest possible example. This is heritability and how we would estimate it. So heritability is basically looking at how predictive a phenotype is from genetics. And in what we call narrow sense heritability, it's how an additive model of genetics is predictive of phenotype. So we write down the simplest linear additive model of the phenotype as a function of genetics. The betas here are the effects of all of the sets on the phenotype. And everything else is what we call environment. I can't fit a model like this because the data is too high dimensional. Even with biobank data, you get too many steps, too few samples. So we have to put some assumptions of the model. So common assumption will be to say that our SNP effects come from some distribution. Details don't matter. There's some mean, there is some variance. The variance is important. It's called a genetic variance. Likewise, the environment has an environmental variance component. Putting all of this together, you have a quantity we call the head. So it's the genetic variance divided by the trait variance. Right? So heritability close to 1 means there's a large genetic component that explains phenotypic variation. It's a very simple model, but it illustrates the key challenges. And just to say, this model can be made more realistic. For example, you can have different variance components depending on whether your genetic variant comes from protein coding regions of the genome or otherwise, by adding these additional variants. And so the goal of this model is we don't care about individual effects of SNPs. They're too noisy, too hard to estimate. Let's just estimate these variance components, and that can tell us in aggregate what is the architecture underlying the scheme. These types of analyses have been very illuminative, also in fluid genetics. So there are multiple studies which try to fit these models of variants thereof to try to understand which parts of the genome might be explaining a lot of trait heritability. Is there enrichment of heritability in particular genomic regions without having to actually identify the individual variant? So the inference problem is to fit a variance component model, the writing a trait as some combination of k variance components for the genetics. So you divide up your genome into k components. Each component, the SNPs there will have some effect sizes that come from its own variance component. And then you have the environment, which has its own variance component. And the goal is to estimate all this from the data. The classical problem, you can try to solve this by computing the maximum likelihood estimator. The challenge is how do we scale this to data? So this scales cubically for most methods. It comes with the sample size, where sample size are hundreds of steps. And this makes these kinds of methods challenging to apply to biobank data sets, although there's been lots of work to try to can make these methods track. So we started thinking about this problem, and we tried to look at alternative ways of estimating it. So in statistics, there are really two classical approaches for estimation. One is maximum likelihood, the other is method of moments. Method of moments has been used to solve this problem in genetics. It goes by the name of Haseman-Elston edge-gene regression. So in method of moments, we'll write down moments of this model. They depend on the model parameters. So here we are looking at the second moment. The second moment depends on the variance components scaled by these matrices. And these matrices, for those of you who come from genetics, they're called GRMs, genetic relationship matrices. which tell you at those set of SNPs what is the pairwise relationship between all individuals. For those of you who come from machine learning statistics, this is often called a kernel matrix. So here we have the population or the model kernels, and now we match it to the sampling. That's what mental is trying. The nice thing about this is this problem has a closed form solution. You can actually figure out what the solution is by solving a linear system. These are the unknowns. These are the coefficients we compute from data. So we can actually just write it without compute the solution without ever having to do any fancy optimization as from the maximum like. But we're still not there in terms of scalability because we have to compute these matrices, these GRMs. And even computing these matrices scales like this. N is the number of samples, and is the number of steps. So again, all of these quantities are hundreds of thousands. So now the question is, could we do better? And that's what got us thinking about this problem. And the key kind of insight is that we have computing all of these very large matrices, but to actually solve this linear system, we only need low-dimensional summaries of the data. We only need traces of these matrices or traces of products of these GRMs. And this is where we use insights from randomized linear algebra, which says we can approximate traces of these large matrices without ever explicitly computing. So we call this marriage of HE regression and randomization, randomized HE, RHE. And for those of you who come from computer science, you can think of it as if you're working with a sketch or a random projection of the genotype. And what that entails is you multiply your genotype matrix with some number of random vectors. So design decision, B of them. The only constraint is random vectors should mean 0 and it decreases. Beyond that, the method is valid for other choices. The smaller the number of random vectors, more efficient it is, the more compressed your data is. So in scales like this, there are some denominators which talk about. But what is less obvious and interesting is you actually get very accurate results for small choices in random vectors. So B can be as small as 10, and you can still get accurate estimates. Another thing which also matters is this method is actually a streaming method, which means you never have to feed in these large matrices, so it's very memory efficient. So this is just showing that compared to maximum likelihood estimation, this method doesn't lose much. You get slightly wider confidence intervals, 10% wider, but on average, it's actually quite close to MLE. On small data set where you could actually do maximum likelihood. And actually, you can also quantify how much randomization adds to your error bars. If you're using 10 random vectors, it's about 25% of the error bars is due to randomness. If you're at 100, it's about 10%. So quickly diminish. And here we compare it to other methods that have been developed in the field for heritability estimation. So RHE is quite accurate compared to these other summary statistic-based methods. The reason for this is they make approximations of different kinds, which don't hold obvious. And the key other aspect is the runtime, when compared to maximum likelihood estimate approaches. So you can actually run this on biobank sets very efficiently. They can also make these models very rich in terms of the number of variance components we're fitting, and that is all scale. I won't say too much about this. You can actually apply it to UK Biobank to estimate heritability. We find additional heritability from low-frequency SNPs and get some additional precision. But what I want to say is this was really, what was interesting for me is this approach is a fairly general approach to fitting more complex models that go beyond linear models. So the basic principle is we write down the method of moments equation and we apply this trace or random projections idea. So we call it randomized with moments. And we are then able to use that to estimate other more interesting models beyond what has been typically considered in the literature. So I'll tell you maybe a couple of quick bottom lines about what we find from this. So the first model we considered, first kind of simplest non-additive model, is a model of dominance. And so I won't say too much about what dominance is, but you can now fit a model where you have additive effects as before and you have a dominance effect. And the question we're asking is there evidence for a contribution of these dominance effects to complex traits? So in a data set like the UK pipeline, we looked at about 50 traits, quantitative traits, in up to 300,000 unrelated by privileging. And we're able to fit them using this framework. And what we find is across these traits, there is substantial evidence for substantial additive heritability across these traits, but almost no evidence for dominance. So there is no statistically significant dominance effect that we are able to detect. And we did some calculation, and if there exists, it's less than 1% in terms of how much it explains trait variance. Okay, so that's about dominance. What about other nonlinear effects? Another possible effect in nonlinearity comes from the fact that there are interactions between genes and environment, so G by E. Well, this is an example of, say, smoking status modulating genetic effects on, say, BMI. So is there evidence for these types of effects in our data? So you can take this basic model and try to fit it to datasets like the UK Biobank. So we have the additive component. And now the way we incorporate G by E is the following. So you have an environmental variable, single one for simplicity. You form all pairs of interactions between the environment and all of your SNPs. And we're asking in aggregate, these all pairs of gene environment variables, that they explain trait variation. So that is quantified by what we call the G by E variance. Again, you can all fit them within the same basic framework. There are some subtleties which was talked about in the presentation today afternoon, which I won't talk about, which is making sure it's calibrated, it has reasonable power, and it scales to large sample sizes. But instead, I'll jump to some results. So we applied the method, we call it Genie, to, again, 50 traits in the UK Biobank, quantitative traits. And in this case, I'm showing 3 environments: sex, smoking, statin receipt, which is medication. And we can ask, across all of these trait E pairs, do we find evidence for G by E? What we find is there is quite a bit of G by E heritability, so trait variation explained by these effects. It's smaller than additive effects. However, it is substantial, so 7 to 15%, depending on what set of SNPs, whether it's array or imperial SNPs we are connected. And here are examples of traits with substantial G by E. Some of these are positive controls, things we would expect, like sex-specific genetic effects on serum testosterone, so that stands out. G by smoking effects on body mass index, this has been reported in the literature. And when you look at G by statin, we find substantial G by statin for LDL, cholesterol, but also actually glucose and HbA1c, which is an interesting story. I can talk about that. They can go beyond genome-wide. In these models, you also have this ability to partition it to ask which regions of the genome might be contributing these signals. This is this enrichment analysis I was talking about. So there are different regions of the genome we could interrogate and ask, where does this G by E signal come from? And interestingly, and this is a hypothesis, do the additive and the G by E signals come from the same place? Do they overlap? So here is an analysis which is quite interesting. We looked at tissues that were measured in GDEX. We looked at genes that are tissue-specific. So that identifies regions of the genome, and we can ask, is there enrichment of additive versus G by E signal in those tissues? So if you look at the plot on the left, when we're looking at BMI, the additive signals are enriched in regions of the genome corresponding to genes that are expressed in brain tissues. So this is prior work. We're just replicating those results. And there's some explanation why this makes sense. When we look at G by smoking and its effect on BMI, we find concordant results with the additives. So this is a case where there is concordance between additive and G by smoking. But now if you look at another trait, in this case, basal metabolic trait, the picture is different. So the additive signal is enriched in tissues which are quite different from the G by smoking. So this is quite interesting because it's telling us that one approach, which is just find the additive effect and then focus on those particular regions of the genome, could be missing regions of action in terms of G by.
Yes.
Okay, that probably of time, but we can also do this for gene-gene interaction. Should I talk about it, or should I happy to?
I think make some arrangements today and share the first stage month. Yeah, I think I'm snapping. I think that's still great to do as you like.
Okay, so we can take the same approach and use this to try to understand gene-gene interactions. Again, this is a very challenging problem in complex trait genetics. There's been a lot of effort. The challenge is you have all of these pairs of SNPs that you have to interrogate. Either your statistical power is low or you have a huge computational problem. So this is where this is work from Lauren and Xiang Jiang. They had this really interesting framework to think about gene-gene interactions. And the framework is basically the following. We're not going to try to identify every pair of variants that affects a trait. Let's ask the question if a particular variant in aggregate across all of the other variants has an effect on the trait. So this is called marginal epistasis because you only are looking at one variant at a time. From our perspective, all that really means is instead of the environmental variable, we are plugging in a SNP here. And now we're asking, is there a variance component that is associated with that is significant? Turns out, again, there's lots to convince ourselves method works. Challenge with G by T is there are many reasons why you might think that you have G by T because of model specification. We have to convince ourselves that those don't impact the calibration of the model. So there are lots of things we have to explore to convince ourselves that the model is still valid in those settings. And finally, it is still scalable. But then we applied it again to the same set of traits in the UK Biobank. And what is interesting is we find about 16 signals, which are genome-wide significant for marginal epistasis for the collection of traits. The challenge now is, again, is this real? So we had to replicate. So we first did a replication within the UK Biobank for this set of marginal epistasis signals. We also took some of these signals which had traits measured in all of us, and we showed that these also replicate in the all of us states. So it looks like these signals are robust. And then we asked, what is the magnitude of these signals? How much of the variance they explain compared to the variance that is explained by just a GWAS. And for many of these signals, it's comparable to how much variance is explained by GWAS. So it's not as though we explain all of heritability or all of missing heritability by these efforts. But when you're trying to interpret a particular variant, these additional marginal epistasis can actually change how we rank or how we quantify the effect of a variant on a trait. And where it lies on the genome, that's super interesting. Most of this, we can partition it. It's actually not on the same chromosome as what we analyze. So it's often with things that are on different chromosomes. And here are some examples. I won't say too much about this, but this is one example where it's a cross-chromosomal interaction for this trait, lipoprotein A. And you can actually, in this case, identify what pairs it's interacting with as significant. And this is what this G by G signal looks like between these two pairs of variants. Here's another example, which is actually really interesting. This was a well-known risk variant for liver disease, which is associated, which is also correlated with this enzyme, which is a marker of liver function. And again, there was a prior paper which had shown that there is some epistatic interactions which are not genome-wide significant. And what we find is when you look at those pairs of SNPs, you do see evidence for G by T. And we think this is a good case for why this method works. You have these individual SNP pairs that are not significant enough to be genome-wide significant. But when you aggregate them by this marginal epistatic approach, you're essentially increasing your power. And so we find several evidences of SNP pairs that are not by themselves significant, but show up in a marginal epistatic scan. Okay, so I think I'm over time. I just talked about all of these questions about what we can learn from about complex trait architecture by applying these methods. I think the key thing for me, at least, is this final point, which is I think a lot of these approaches are now revealing the role of context, either environment or genetics, in how we think about complex traits. But with this, I should thank all of the students and postdocs who led a lot of this work, and happy to take any questions if there's time. That's what that is.
I'm curious what these are phenotype intuition framework is recent now within radical how to us. they argue that consumer types are intended really to use some false types, but how is the work in terms of like that? How the false positive bad and downstream and deep.
Yeah, so that's so PUBG was there is several work in that framework. PUBG was the most recent. The whole kind of thinking is what they call post-prediction inference. So imagine there's going to be a lot of machine learning applied, a lot of phenotypes are machine learning based. How do you make sure that everything you do downstream is valid? And that's the kind of the type of post-prediction inference, including PUBG was. And what is powerful about those approaches is you might have biases in your prediction and interpretation. If you use the PUBG loss approach, then you will still get valid inference downstream. So you can think of it as like a two-stage approach. You have the imputation, and then what you do with the imputed feelings. Now, in our approach, so that's why I said you have to be careful about the validity of your imputed feelings. Right? So there is no guarantee that we get that the phenotypes that we are imputing are going to be valid. So you can do that by employing this type of approach, or by doing additional things that we did in the paper in terms of external replication, which is extremely important. And that falls independent of whether you PUBG or not. It's never clear that you are imputing phenotype that is relevant as opposed to just copying a phenotype which is non-biologically fit.
And if so, another question, one rate that is fit by figure, how does that harmful is sample of overlap and then it's still not a bit better.
Yeah, it's a big question. So here, when we analyze it, we work with unrelated individuals. So that is a step that we took, like started to basically value methods. How it applies, that could bias your results quite a bit. So you have to be careful about sample overlap. Can you account for it in a meaningful way? That's a super interesting question. Because, you might actually be able to learn more accurate phenotypes by leveraging sample related. So that's not explicitly accounted for.
Thank you.
Thanks, Gabby. Really nice talk. I went back to the interesting stuff from your 2020 paper. I was wondering if you called the interest tracks in the West Africans. I guess you didn't show it here, but you mentioned that you did that. I wonder, did you compare the location of them as interest tracks versus, I guess, like genomic annotations? Because I guess, like from the traditional, or not traditional, but traditional duration and regression, we have like some known findings of like we see less archaic ancestry and lower combination rate than say and by B statistic and the gene density. I was just wondering if looking at that could tell you something that maybe about this unknown donor, if did that align with our expectation for non-group populations that it was presumptive ancestry, or if it potentially was some, I know there's been a lot of talk now about these really like deep structure models, and that some people, there's no integration actually. Skit.
Yeah, so we didn't put that analysis in the paper, but there's unpublished work where we have actually done that. So essentially it's doing something like this picture here, right? So you can look at archaic ancestry now is an unknown ghost archaic ancestry and ask whether there is some correlation with different genomic adaptations. So for example, we looked at the B statistic and we see a very similar trend. Now, whether that necessarily means, whether this argues about interpretation versus not, I think that's harder to, that by itself doesn't kind of tell us one way or the other. It does say that there is some kind of, that these were diverged enough that they must have been to find correction on these tags.
Right. Sorry, I wasn't kind of necessarily caught it. That was evidence. I was just trying to, I guess, make the point that it would be really interesting if you look at the facts and it didn't line up with our trends. I guess we see like non-African thing with near qualities in ancestry.
Yes. So it does line up with our case. Although for some reason it did not through the
Writing about this is like what kind of sort classic.
Yeah, so that's a really interesting question. Um, so just to give you a perspective on the marginal epistasis, so so that was UK Biobank analysis said we find about 16 trait, marginal epistatic segments. If you look on the bottom left here, that's UK Biobank, and that was we are doing a discovery kind of analysis where we look at half the UK Biobank, and you'll see that the number has shot.
Right.
So that's again still hundreds of thousands in that plot. And similarly, when we go to all of us, where it's about 50,000, you'll find that everything is closer. So my guess is, yeah, anything below 50,000, it's very unlikely. I mean, it's hard to be able to find evidence, although some of these might be strong. Some of these are very strong signals, but yeah, that's probably the sample size really to compute.
And based on that, the point on infrastructure that the missing grants. So anyway, that it is in the trafficking, you said that there are ways to import, like learn about the structure, everything that's in the data, and then use that sort of maximum for the identification. Do you think that this sort of, like, is this one in the maps?
Yeah, I guess that would be taking into its logical intuition, where you have a model that learns mass simultaneously as it's doing imputation. Again, there are subtleties there because if you talk about more complex signals like MNR and so on, those are really hard to learn just from data. Like I think there you would need to have actual kind of assumptions baked in, which will say that this is what the mask should be like. But there might be other settings where I think the models would definitely learn the mask. And this is an approach that people have actually tried. We haven't tried it, which is to give the model as input both a mask matrix and a observed matrix. Right? So you have two matrices, and you let the model figure out what's the relationship between them. Well, in our experience, at least those very simple approaches haven't worked well. But no, I think there's more to be done here in terms of really thinking carefully about how do you get the model to learn this. And maybe I'll say one more thing because this is working in this field. We've also been looking at the ML literature, which has tried to do some of this, and we find that there is actually a very big kind of gap between, say, the biobank and the genetics literature and what people think about tools versus what people do in the ML literature. For example, as far as we are aware, we are writing paper, but this structured missingness has not really been at kind of the way people think about doing imputation there. Oftentimes, even the benchmarks reflect that. So they take completely observed data and they would put some simplistic models to simulate missingness. And what we actually observe is when you take state-of-the-art ML models from last year's NeurIPS and so on, and you apply them to UK Biobank, they do much worse on these data sets which have realistic missingness patterns. How much of that is maybe there's some real kind of tuning of these more complex models versus them not being tuned to actual missingness patterns is not super clear. But I also think there is kind of a gap between these two narratives.
We've had a bit more similar thing rendering the patient, it's elsewhere that someone made this thing that just because they're clear on based on these data assumptions of them that I start already fast so fair to the actual between
and indicators.
Yeah, so I guess that's an con.
So, I can tell you that, right? So I assume, and left that, bring people in. We kind of have to take down this structure, I guess, from phenotypes that it's not nothing to mention that there's relations between phenotypes. Bring this into it.
I think that's, I guess, That's what it is. So with correlations between graphene clips is we're not using genetics in this analysis. Again, that's another potentially the same section in pursuing.
But for each benchmark, like a tissue and a something, I look up a benchmark there to be a base. You're like this day, like the bodies in they work the food structure. It's not, I don't know, like,
Yeah, I mean, I think, I don't haven't followed the latest computation efforts, but like whether people are using
stem sample like the students, right? It's a
you got the best. Yeah, so I guess that could be an example where those may not be the connected to pattern that you expect to see in the data. So, yeah, I guess the other maybe difference, it depends on what type of genetic computation you're doing, whether it's array to per genome or was it low coverage to whole genome. Because again, those are just doing the sequence pattern together. So maybe the second is more relevant for picture period.
and but thank you. But and I think I've seen that from somebody to share it. Because we're gonna want to do with I just want to frame.
That was, I mean, so, what I heard, I mean, helps like, you don't get text like that, do you? No. Okay. That was like something I was worried about, and
I think I might
just went back and forth. That do it, right?
yes,
State side.
there is mistakes.
Right, clearing up of it.
I can
Why is it accent buts in shit?
So,
I don't even see the cut up and the box questions. Like wait on me. Did that for to start too long the current the wrist on the request. So yeah, that's the mistake that does that in there. So the like in the top there.
So it depends on whatever you can give as for. So as long as I need to say about the shit that's it's at DSE, we should get they're not going to be it's solving the same kind.
And then
So it's an input method to method it was our case of if they call it those cases of bag. See what I then
sure it's happy. But I tell you but if there's basically and then we're sorry I couldn't
wait when we can
but what's okay according to but it's not.